Hi there. We are going to dive into chapter 11, meiosis and sexual reproduction. So, meiosis is the production of gametes, which allows for sexual reproduction to happen. Gametes are eggs and sperm. So, meiosis is a little bit different than mitosis in that it has two phases, but they're only really unique stuff in meiosis 1. Meiosis 2 is identical to mitosis. Yay! Okay, so you're going to need to know the differences between meiosis and mitosis and how we get genetic variation. So, asexual reproduction is just mitosis. It's just making copies of cells. So these are identical, genetically identical daughter cells. They're basically clones. Same DNA, same number of chromosomes, same genetic information, same DNA sequences. Everything is the same. Meiosis is different. We get variation. Okay, this is where we make our gametes. Again, our gametes are our eggs and our sperm. Okay, these are the only cells in our body that don't have the same amount of DNA as all the other body cells. Okay, so these are our sex cells, gametes, eggs, and sperm. All mean the same thing. Okay, they are haploid. Haplo means single or half. So they have half the amount of DNA that a normal cell has. So a normal body cell has 46 chromosomes in it. A sex cell has 23. Why? Well, eventually we're going to smash an egg and a sperm together. And when we do that, we don't want too many chromosomes. So if egg has 23, sperm has 23, and we come together, we have 46 and we're good. So diploid, okay, di means two. Cells produced from mitosis are genetically identical and diploid, whereas cells produced from meiosis are genetically unique and haploid. So, little review of what homologous chromosomes means. Okay, these are paired chromosomes that carry matching genes, meaning they control the same inherited characteristics. Okay, so homo means same, okay, and the ending logos actually means information. So it literally means same info. Okay, so if we look at this little diagram here, we have this blue chromosome on the left from dad and this red chromosome on the right from mom, and they carry genes for the same traits in the same place on the same chromosome, except they have different expressions or different copies of their genes, right? So mom has blonde hair, whereas dad has brown hair. Mom has blue eyes, whereas dad has green eyes, okay? So our homologous chromosomes are carrying the same inherited characteristics in the same places on the genes. So when we prepare for mitosis, okay, so we're going to go over some vocab to just review, okay? We have to duplicate during S phase. We have to duplicate our chromosomes, all right? And so when we're undergoing mitosis, we duplicate our chromosomes and we end up with sister chromatids like this here on the left, all right? This is one duplicated chromosome. What we're going to be dealing with in meiosis is the pair of homologous chromosomes, because remember, we've got a maternal set from mom and a paternal set in blue from dad, all right? So our pairs of homologous chromosomes are going to be what we're dealing with, as opposed to a simple one sister chromatid. Here we have a pair of sister chromatids, one from mom, one from dad. Now, as we prepare for meiosis, you need to know this, okay? These pairs of homologous chromosomes undergo synapsis, okay? It's a term you need to know. And when they do that, they line up gene by gene and form what's called a tetrad. Okay, tetra means 4. So think it's just like 1, 2, 3, 4. Okay. And this is where they're preparing to exchange genetic material during the process of crossing over, which is where we get genetic variation. Okay. So here, okay, in prophase 1 of meiosis, we have the centrosome and spindle fiber forming. We have the nuclear membrane breaking down, the nucleolus disappearing, and then we have synapsis that's going to occur where our homologous chromosomes are going to pair up, and then we're going to form that tetrad, which is our homologous chromosomes pairing up. And then crossing over is going to occur where they're going to kind of dance and they're going to exchange genetic material. And this is where we get genetic variation that allows us to have different responses to stimuli in the environment. In metaphase 1 of meiosis, your tetrads or pairs of homologous chromosomes are going to line up along the metaphase plate, okay, attached to microtubules. And then in anaphase 1 of meiosis, your sister chromatids themselves, the whole sister chromatid, is going to be pulled up towards the pole of the cell. Okay, so this is a difference between mitosis and meiosis, where we have the pairs of homologous chromosomes separating, and they separate randomly. This is called independent assortment, which we'll get to. Then we have telophase 1 and cytokinesis, where nuclear envelope reforms, nucleolus comes back, chromosomes relax, and cytokinesis begins. So overall, prophase 1, nuclear envelope disappears, nucleolus disappears, chromosomes condense, homologous chromosomes undergo synapsis to form a tetrad. During that, while they're in the tetrad, they undergo crossing over, which is when they touch each other and exchange genetic material. Okay, that's prophase 1. Also, our spindle fiber and centrosome shows up. Metaphase 1, our pairs of homologous chromosomes or tetrads line up at the metaphase plate. And in anaphase 1, the pairs of homologous chromosomes are pulled apart up towards the poles of the cell. Okay, the whole sister chromatids remain attached. Then we undergo telophase 1 and cytokinesis. Then we have meiosis 2, identical to mitosis. Okay, and then what we end up with is 4 unique daughter cells that are haploid or have 23 chromosomes, or half the amount of chromosomes that a normal body cell has. So mitosis gives us 2 diploid, okay, full set of chromosomes, identical cells. Meiosis gives us 4 haploid unique cells. We get genetic variation from crossing over, which is when our pairs of homologous chromosomes form a tetrad during synapsis, which is synapsis is when they come together. Then they're in the tetrad. Then they dance and touch each other during crossing over and exchange genes. Okay, we also have independent assortment, okay, which is when the chromosomes line up at the metaphase plate, they line up randomly, okay? And so you can get millions of different genetic combinations from this random assortment. Okay, so these are just 2 possibilities where you see here, it's entirely possible for one gamete to end up with all of one parent's genetic material. Like, my nephew is identical to me, and I guarantee he is more closely related to me than he is my sister, most likely, because he has so much of my genetic material to look like me. And so there are many different combinations that are possible through independent assortment, okay? And so we're talking about like 2 to the power of the 23rd, so 8 billion, a lot. That's a lot. Okay. And then we also have random fertilization. So, mating in the animal kingdom is complex. Sexual and social behavior is very complex. And so, you never know who is going to mate with who. And so, this is very random as well. Or not really super random. There's, different. Sexual selection is a whole thing in and of itself that we are not going to get into during this course. However, it does contribute to fertilization. Anyways, lots of possibilities. That is meiosis in a nutshell. Have a great day.